Good afternoon and welcome, everyone. This is a TEAS test prep session over genetics and Mendel's laws. If you've participated in our webinars in the past, you know that we are currently doing a two-part series where we have content on one day and then we have a practice test on the next day. So today, it's all about content, learning genetics and Mendel's laws. And then tomorrow, same time, 3 p.m. Eastern Standard Time, we will have a practice test, very much set up like the TEAS, where you will have an opportunity to put in the chat your responses. And we will talk through the rationale, why things are correct, why things are incorrect, and really set you up for success on the TEAS and specifically focusing on Mendel and genetics. So please join us tomorrow and welcome today. This does cover the sections S.2 and S.2, and that is protein synthesis, genetics, all of Mendel's stuff like Punnett squares. So we got a lot of good stuff coming at you. So sit back, enjoy. Before we get started, though, I do like to share with you all of the amazing resources that we have available to you at Archer Review. I feel so blessed that we have so many wonderful things at our fingertips for you to use in preparation for the TEAS. I know that I've communicated with some of you, and many of you are preparing and studying hard to take that TEAS this summer. And so we have all of these wonderful resources for you. Our first one that I always love to share on the first day of our two-part series is our question bank. Our question bank is so amazing. And so I do want to just share that with you real quick because it is out of this world, just to be honest with you. It is unlike any other. So let's take a look at our question bank. So our question bank, one of the things that I love most about it is that you can tailor it. You can tailor it to whatever you feel you need to focus your attention on. You can create your own test, as I'm going to do right here. And when I create my own test, I do have the option to do the tutorial version or the timed version. Now, the tutorial version, I like when I go through it, maybe the first time when I'm still really honing and practicing my skills, because I'm going to show you that one. It's going to give you the rationales. It's going to explain why an answer is the correct answer. And even in most cases, link you to a on-demand video that can talk you through that content even deeper. But I would say as you get closer to your TEAS, do the timed version, because the timed version will help mimic that pressure that you're under when you're taking it in real life. And I think that a lot of us struggle with test anxiety, and that is one way to get comfortable with the anxious feeling of I'm timed while I do this. So at this time, then you would pick your question types, unused. I'm going to just go with all. And notice we have 2,040 questions in our question bank. I love it. You can pick how many questions you want per test and then choose your subject. So since we are in our genetics and Mendel laws, I'm going to choose biology. And then it tells me, okay, well, which categories in genetics do we want? I'm going to choose the two areas that we've got today that we're focusing on, and it's going to build our test. Now, I'm not going to even read through it. I want to get to content, but I wanted to just give you an idea. So I'm going to just click an answer, didn't even read it. And we can see that was incorrect. It tells us how many people have chosen that answer. It tells us what percentage of people are getting this one correct, and so on. It tells us why answer A is the correct choice. It goes over why the others are incorrect. But here's the other thing that I wanted to show you. You know, time is of such an essence for all of us. They say time is money, and boy, isn't that the truth. And so what I love about this is we have a video all about Mendel's laws of inheritance. But notice that that video is 53 minutes long. That's a long video. You may not have 53 minutes to figure out why you don't understand this question, but it'll take you right to a topic to support this question. So you can see, you can go into the video at 17 minutes and watch it for about 5 minutes, and that helps you then get a deeper understanding of that question. And that is just one of my favorite things about our question bank. So definitely wanted to share that with you. Now, we also have that on-demand video. I kind of gave you a clue of that as I talked about how we have this 53-minute video, but that you could go to the time-stamped spot. Well, now tomorrow when you come back for our practice test, I'll show you the cool stuff that's within our on-demand videos. They are so awesome because not only do you get the on-demand deep content, but with those you get notes and cheat sheets, and our cheat sheets are beautiful. So I will definitely show that all to you tomorrow. But I like to break it up. Question bank one day, on-demand videos the next. Next, I wanted to mention that we do have tutoring. So if you need a little bit of personalized assistance, either in a small group or individually, Tammy can help you out with English and reading. I'm your science person, and then Brandon Kraft is your math person. So you can scan those QR codes, or you can reach out to us via Archer Review's website and schedule a tutoring session. So keep that in mind. As you know, because you're here, we have free live webinars. That's what you're at today. And we've got a lot going on right now. I think we have like 9 webinars all in a row. I know I've got 4 of them this week. So I have the content and the practice test today and tomorrow for genetics and Mendel laws. And then Thursday and Friday, we have scientific reasoning, same time. So come back for those as well. You can scan this QR code here and rewatch old webinars. And then, of course, we'd be remiss if we didn't offer some bundles and some ways to save a little bit of money. So we have the Sure Pass and the Sure Pass Pro. The difference between those is that the Sure Pass gives you pretty much everything, your Q bank, those on-demand videos with the notes and the cheat sheets. But you don't get a tutoring session until you jump up to the Sure Pass Pro. So if you're thinking you need tutoring in any of the topics, go for the Sure Pass Pro, and that will offer those tutoring options for you. Also, we've been doing something pretty cool on our social media in that we highlight a topic a week and then we really dive deeply in it and give you like 2 mini lessons and 2 practice questions. So this week was chemistry, and we talked all about acids and bases and buffers. So if you are not following us on social media, definitely do so. Obviously, that's all free. And so it's a free, and they're short little tidbits. You know, a lot of times, again, time is so important, and you're going to get a short little tidbit that's maybe 4 minutes long and help refresh or help deepen your understanding of those topics. So check us out, TikTok, Insta, Facebook. We're there with all of those topics helping you out. Lastly, if you need something, if you want a tutoring appointment and it doesn't seem to line up with the schedule that is available on our website, definitely email us at prenursing@archerreview.com. We can often work with you, come up with a time that, even though it's not on our website, that one of the tutors might be able to move some things around and become available. So keep all of that in mind and reach out. All right, folks. So as we said, we are going to focus on genetic material and proteins and Mendel's laws of inheritance. So without further ado, we're going to get started. So the first thing that we really have to focus on when we talk about genetics and inheritance and Mendel's laws is DNA. Right? DNA is the basis for all of that. So we have DNA and we have RNA, and they are essential nucleic acids. If you were in my macromolecules session last week, you remember that we have 4 main categories of macromolecules, and nucleic acids is one of them. So DNA and RNA are vital nucleic acids, and they play a key role in genetic information and in protein production. And they're going to work together to ensure that the traits are passed on to offspring and that proteins are built. So DNA is serving as the recipe for us, and RNA is serving as a tool or a messenger for protein production. So one of the things that's really important for us to understand is that DNA is responsible for our traits. So if we break it down and we remember that the nucleus is the central region of our cells and it houses the DNA, and DNA will coil up tightly into chromosomes. Chromosomes are made of DNA that is wrapped around histone proteins to help with its structure. On a chromosome, a segment of it, such as this box we have right here, represents a gene. And a gene is essentially the recipe for a particular trait, like blonde hair, blue eyes, the ability to roll your tongue, the ability to dribble a basketball better than someone else. All of these traits that we have that make us who we are reside on our chromosomes in genes. Now, genes, how they make those traits is that they have the instructions to make proteins, and it's the proteins that determine those traits: the blonde hair, the blue eyes, the ability to roll the tongue, that sort of thing. So now let's shine our spotlight a little deeper into DNA and RNA before we go into protein synthesis, since proteins are really where it's at. So DNA is considered a double helix. What does that mean? It means this round structure. It's called the double helix, and it's a twisted ladder shape where the sides are made of a sugar and a phosphate. And that's what we see all along here. That's a phosphate, that's a sugar. Phosphate, sugar, and so on. The sugar is deoxyribose, and the phosphate is just called a phosphate or a phosphate group. Now, the steps of the ladder are often called rungs, and I know that that's a word that's not familiar to everyone, but it's just the steps of the ladder. And the steps of the ladder are those bases that you're probably familiar with: the adenine pairing with thymine, the guanine pairing with cytosine, or as we typically write it, A pairing with T, G pairing with C. Okay. Now, a DNA structure to make this twisted ladder shape is actually made of monomers known as nucleotides. And then those nucleotides fit together kind of like puzzle pieces in order to make that spiral shape. So a nucleotide, which we have pictured right here, is composed of three things: the phosphate group, that's what's along the side of the ladder; a sugar, that's also in the ladder; and then a nitrogenous base. The nitrogenous base is the adenine or thymine or guanine or cytosine. Now let's turn our attention to RNA. RNA is still a nucleotide, much like DNA, but notice it's half of a spiral. It's not a complete spiral. They say that that's a single-stranded molecule. Now, another difference is, remember that DNA has thymine as one of its bases. RNA has uracil instead of thymine. So that means if you had an adenine in DNA and you were pairing it with an RNA, you would have to pair it with a uracil instead of a thymine. Other variations about RNA is that the sugar that is part of that side or that backbone of the structure is ribose, not deoxyribose. That's where RNA gets its name, ribonucleic acid. R stands for ribose. Like DNA is deoxyribose nucleic acid, okay? Now, RNA functions as a tool in protein synthesis. It's going to be a messenger, it's going to be a tool. It's essentially a multifaceted molecule that's offering several jobs to assist with protein being built. Two key forms that are involved in this process are mRNA, messenger RNA, and tRNA, or transfer RNA. Now that we've laid the groundwork for DNA and RNA, let's take a look at protein synthesis. So protein synthesis is all about taking the information that's in DNA and using that to build proteins. This is a two-step process. Our first step is transcription. Transcription is going to occur in the nucleus, and this is where the DNA code is being copied into mRNA. After transcription, we have translation. Translation occurs in the cytoplasm on the ribosome. And in this case, that mRNA sequence then is read and the protein is formed. Okay, now we're going to break this down step by step. All right, transcription. Here's the big idea before we go through what's on the screen. DNA is enclosed in the nucleus. DNA cannot leave the nucleus. It's not environmentally, I shouldn't say environmentally, it's not to any evolutionary advantage for DNA to leave the nucleus. It is protected. It is doubly protected because we have our cell membrane and then we have our nuclear membrane. So DNA is very well protected in there. And so we don't want it to leave the nucleus. But we have a problem. We have the instructions on DNA to build a protein, and the protein has to be built on the ribosome out in the cytoplasm. So essentially, we have to solve this problem of how do we get the recipe for that protein from the nucleus out to the ribosome? And that's our first step of transcription. So in transcription, what happens is we're kind of like making a photocopy of the particular instructions we need so we can take it out to the ribosome. So how does that work? Well, you might guess, because enzymes are so beneficial and so helpful in our body, this starts with an enzyme. It starts with RNA polymerase. RNA polymerase is going to guide the production of mRNA by binding to and unzipping the DNA and using the strand as a template to create the photocopy, so to speak. So it unwinds, it breaks the bonds between the A's and T's and C's and G's, and then it starts building essentially a photocopy. And that's what we see right here. Here's our enzyme, and it's building that copy or that mRNA right there. So if it's an A on DNA, it's pairing with a U on RNA. If it's a C on DNA, it's pairing with a G on RNA. Now, once mRNA is built, then we have to go on to translation. Translation is where that recipe is going to be read and the protein can be built. So that's our next step. So the mRNA, that photocopy, so to speak, leaves the nucleus, travels out into the cytoplasm, and then lays on the ribosome. That's what we see illustrated right here. Okay. And so now, essentially, this is like you've got your recipe, and your recipe is a protein. And so now we have to go through grabbing all of our ingredients and putting it together. And that's what's happening in translation. So mRNA lays out like a recipe book. And this is where tRNA comes in. tRNA is a delivery mechanism. It's a delivery molecule. It's bringing over the ingredients or the amino acids, because those are our monomers of proteins, to the ribosome in order for that protein to be built. Now, it follows the recipe just like we would if we were building a protein in our kitchen by reading it. And it reads it in groups of 3 called codons. Okay, so the mRNA is read in groups of 3 called codons. And the tRNA knows it's got the right amino acid because on the bottom of the tRNA structure is an anticodon. And so they line up. So if the mRNA was AUG, our tRNA would be UAC. And as long as that tRNA has the right match, it's like, yep, we're good. I've got the right amino acid. I'm going to drop it off and go get another. And so then at the ribosome, those amino acids are being linked together, forming a polypeptide chain that will ultimately become a protein. The polypeptide chain will form at the ribosome, and then ultimately, once it's done, it's going to head out to other organelles, such as the endoplasmic reticulum, in order to fold it into a functional protein. Okay? All right, I got a question here. Let's take a look at our question. So the complementary, someone's asking about complementary DNA, mRNA. What we've got going on here is we're building RNA. We're not building DNA, okay? We're building RNA. And so mRNA is the photocopy, the complement to that part of the DNA, okay? That's what we built in the nucleus. And then that is our photocopy that tells us how to build our protein. Okay, hopefully that answers your question. If not, feel free to ask it again. I'm happy to help. All right, so now we are oh, I'm sorry, it skipped. No, we're good. Now we are transitioning into mutations, okay? Mutations are mistakes, okay? They are mistakes actually during DNA replication, something we went over last week during one of our webinars. And that mutation occurs and it's fine, maybe, until it needs to be read to build a protein, and then we have problems. And that's what we're going to take a look at next. Now, mutations can be caused by environmental factors, and they can also be caused just by a mistake in DNA replication. That's really what it boils down to. So it impacts protein structure because mRNA makes its photocopy, so to speak, based on DNA. So if the DNA has a mistake in it, it's going to copy that mistake. Okay. And so that's why it's such an issue. So now mutations that we'll focus on today will be deletion, insertion, and substitution. These are all DNA mutations. There are other forms of mutations that occur on like as a chromosome whole, but our focus today will be deletion, insertion, and substitution. Now, one of the beauties of them is that they're very much like they sound, which is fantastic. Another thing that we want to point out is that actually most mutations are harmless. And even some of them we have during our cell cycle, we have checks and balances where our body checks for mutations and fixes them. But some can cause health complications. So if they get past a repair mechanism, then we can lead to things like cancer or genetic disorders. All right, so our first type of mutation is a deletion mutation. And I love this, like I said, because it's exactly what it sounds like. It's a part of the DNA gets deleted. It's simple as that. And it could be 1, 2, 3, 4. It could be a multitude of bases. There is no set number. And that's what we see in our illustration here. We start off with A, G, C, G, T, and then we have a problem right here. Like this is no longer present down there. And we can see that we've got a chunk that has been cut out right here. That's all been cut out. Now, why this is a problem, if we remember that our mRNA is read in groups of 3. And so when we have this chunk cut out, it's generally not a chunk in groups of 3. And so it causes the read of the mRNA to shift. They call that a frame shift. And that can lead to the protein not really being formed properly. And so then it doesn't function properly. Or it can stop the recipe, the building of the protein too soon. And either way, we end up with a protein that's just not functional. And this can lead to genetic disorders like cystic fibrosis. Now, another one, again, I love it because it's just like it sounds, insertion mutation, is where a chunk of DNA is just kind of randomly inserted into our structure or into our DNA structure. And in that case, same idea. We can end up with a abnormal protein, a non-functional protein. We can have a frame shift because, for example, see there, it was a chunk of 4 bases. Well, everything's read in groups of 3. So now it's shifted everything after that. All of that would be read differently because we had a group of 4 thrown in there. So this leads to developmental disorders, and this can also be what leading to Huntington's disease. So someone asked, what do I mean by frame shift? Well, let's say that we had cat ate dog. Let's say that that was our DNA because we want it to make sense. And then a random word got thrown in there. Cat as ate dog. But if we're reading it in groups of 3, we're good here. That would be our normal codon. But now it would be cat as a Ted og. See how it doesn't make sense? And that's essentially what the amino acids it would make no sense for the tRNA. The tRNA would come over, this one would be okay, but then this would be a mess, this would be a mess, and it wouldn't even know what to do with only 2 bases when it's used to reading 3. So that's the problem. It messes everything up downstream for that protein, and that's why they call it a frame shift. Good questions. All right, our next one, substitution. I love this. This is just like all the others where it's what it sounds like. But in this case, it's only 1 nucleotide. So we're reading this one all along, all is well, and then we initially had an A, and now all of a sudden we have a C. Now with substitutions, they can result in 3 different types of mutations. A silent mutation where it actually doesn't change the amino acid because there are several words or 3 groups of codons. There are 3 groups of codons that stand for the same amino acid. You can kind of think of those codons as synonyms for the same amino acid. And so sometimes the silent mutation comes along, it doesn't change anything because it still codes for the same amino acid. Then we could have a missense mutation where a different amino acid is incorporated because it did code for a different amino acid. And that may or may not impact the protein. It depends on what amino acid is incorporated. And then a nonsense mutation. This is where a stop codon is introduced and it stops the production of the protein. So I've been talking about how mRNA is like a photocopy of the protein recipe. And ultimately, there is a start codon that says, hey, the recipe starts here. And there's a stop codon that says, hey, the recipe ends here. And so a nonsense mutation might throw in that stop codon in the middle of the protein production. And that's what that means. All right. So with substitution types of mutations, you would typically think, well, it's only one nucleotide. It may not cause that big of a deal. But in some cases, it can actually range to a pretty severe case. For example, it's a single nucleotide substitution that causes sickle cell anemia. And that's, pretty significant response to one nucleotide being altered. All right, we got some more questions. Silent mutation. No. So someone asked, a great question. A silent mutation means no change, so no mutation happened. No, a mutation happened, just like we see in our visual right here. But there are several different codons that code for the same amino acid. And so essentially, it's the same word, so to speak, that's saying bring lysine over. There might be 3 codons that call for lysine. And so it just so happened that even though that letter changed, it's still calling for lysine. And so it doesn't change the protein that's being built. All right, we are through our first section and we are on to the laws of inheritance. This is focusing on Mendel's laws. So if you're not familiar, Mendel was a monk in the 1800s, and he was very interested in how things were inherited. And he worked with pea plants all his life, crossbreeding them and studying them, and he came up with laws, and he came up with a lot of how we understand how things are inherited. And so now we have a little bit more sophisticated language for it because we know about DNA, but at the time he didn't know about DNA. And so it's pretty amazing, actually, that he was able to figure out what he was able to figure out without even having DNA knowledge. So what we now know from Mendel's laws, we've got our first one here. This is Mendel's law of dominance, that traits that are inherited are determined by our genes. Remember, we talked about that. Genes reside on the chromosome. But each gene can have an alternate form, and this is known as an allele. And when we inherit our alleles from our parents, a dominant one will be the one we see. A recessive one will only be expressed if there's not a dominant one there. Now we're going to break this down, so don't panic if that still doesn't make sense to you, because this is where language that maybe you're familiar with, genotype, phenotype, homozygous, heterozygous, come from, okay? So genotype. Genotype will be the set of genes or alleles responsible for the trait. So this is where we have big B, big B, big B, little b, or little b, little b. We use letters to represent a genotype. And this is where a capital letter and a lowercase letter, such as big B and little b here, are going to represent different versions of the gene or the alleles for the gene. Now, a great way to remember genotype versus phenotype, because we're going to talk about phenotype in a minute, is that geno starts with gene and is representing the genetic combination that we're studying. All right, so let geno and genetic remind you that it's standing for the alleles. It's standing for the underlying genes. Now, phenotype is the expressed physical characteristic that we are going to see, such as hair color, eye color, certain diseases. Okay, so let the phenotype, pH, physical pH, remind you that this is the physical characteristic that you're going to see. Okay. All right. So I'm getting a question here about an order, DNA, gene, chromosome, genome. So DNA spirals up into chromosomes. A segment of the chromosome is a gene. The genome is the entire human recipe of DNA that they initially developed, I think, in the 90s. So hopefully that helps. All right. Now, we were talking about this idea of law of dominance. This is what all of this language comes from, alleles and genotype and phenotype. And when we talk about the law of dominance, we have then some more language here. We have alleles that would be homozygous dominant. This means that you would inherit 2 dominant alleles for a gene. Let's say big B, big B. And that would be referred to as homozygous dominant. Homo meaning same, and dominant because we have the capital letters. Now we have homozygous recessive. This is where you could inherit 2 of the recessive alleles for a gene, such as right here. Homo meaning same, you have 2 of the same alleles. Recessive being the trait that's only going to show up if there's not a dominant trait there. That's the whole idea of Mendel's law of dominance. Now, there is another form too, heterozygous. This is where you could get one of each. You could get a capital and a lowercase. You could get a dominant and a recessive. In this case, because of Mendel's law of dominance, it means that you'd only see whatever this stood for. So if big B stood for brown eyes, even though you got the little b, let's say, stood for blue eyes, you'd see brown eyes because the dominant trait overpowers the recessive trait, and we only see the dominant trait. That is Mendel's law of dominance. Okay. All right. So now we have another law. Mendel has 3 laws that we have to be aware of for the TEAS. In this law, the law of segregation, we learn that each organism has 2 alleles for a trait, just like we had big B, little b, or little b, little b. In this case, we have examples using F. And these alleles separate when we form our sex cells, our gametes, in meiosis. So this would be like your normal chromosome, but if you're a female, this would be a potential egg. This would be another potential egg. If you're male, that could be a potential sperm. That could be a potential sperm. Then, when the sperm meets the egg and those unite, we have an allele from mom, we have an allele for dad, and we have the right number of chromosomes. So this ties into meiosis if you participated in my webinar last week, because remember, in meiosis, we have an original cell with 46, but we end up with ones that are 23 and 23. And that is because when the sperm meets the egg, then we have the right number of chromosomes, 46. This all ties in because you got one of your alleles here, let's say from dad, one of your alleles from mom. In this case, it would be a heterozygous version. And now the resulting zygote would be heterozygous for whatever trait we're studying. So I'm going to ask what homologous means. Homologous refers to the pair of alleles that are on the same chromosome. So, for example, these are homologous alleles. I often say they kind of remind me of a pair of shoes because they're not identical. Your left shoe is slightly different than your right shoe, so that would account for a big B and a little b, but they're still the same pair of shoes. Homologous alleles are referencing the same trait, and they're nearly identical with a slight variation. So if we're talking about eye color, they're both representing eye color, but one's going to represent brown, the other one's going to represent blue, or whatever shade that is inherited. All right, so because Mendel has this idea of the law of dominance, and because he has this idea of the law of segregation, we can actually use a Punnett square to predict the probability of acquiring a certain trait. So for example, in humans, freckles are dominant, and the absence of freckles is recessive. It's telling us to perform a monohybrid cross between 2 heterozygous individuals. What does that mean? Well, a monohybrid cross is that we're just looking at one trait, freckles. We have two heterozygous individuals. So that means that the male has the potential to donate a capital F and a lowercase f, and the female has the potential to donate a capital F and a lowercase f. So let's take a look at how this works. There's the potential, this could be the male. It could be the female too, but let's say that's one possible sperm. That's another possible sperm. This is one possible egg from the female, and this is another possible egg from the female. We bring them into our Punnett square. See how we brought one down, we brought the other over. We bring it into the Punnett square, one over, one down, one down, one over, one down, one over. Okay? Now we have to decipher that. What does that mean? Well, we would report genotype and phenotype. Okay? So in this case, our genotype, we would have the 1 out of 4 would be the dominant, homozygous dominant. 2 out of 4, heterozygous. 1 out of 4, homozygous recessive. Now, this does not mean, this is very important, this is determining probability. This does not mean if you have 4 kids that one's going to have a dominant trait, 2 are going to be heterozygous, and one's going to have the recessive trait. No, it means we have a probability. We have a 1 out of 4 probability of getting the homozygous dominant trait. We have a 2 out of 4 probability of having heterozygous genotype. We have a 1 out of 4 probability of having the homozygous recessive genotype. Now, what will they look like? Well, we have to remember Mendel's law of dominance here. 3 out of 4 will have freckles. Why is that? Well, because every single one that has a capital F would show the dominant trait. So there's a capital F, there's a capital F, there's a capital F. So 3 out of 4 is our probability that we would have freckles. 1 out of 4 is our probability that we have no freckles. Now, we could report this also in percentages. It'd be easier to say that we have a 75% probability of having freckles and a 25% probability of no freckles, okay? But remember that this is probability, not a recipe for if you have 4 kids, how are they going to turn out? Okay? Now, Mendel's got another law, our third and final law, that states genes from different traits are inherited independently of each other. Now, I think that this is probably the most difficult one to make sense of. So what he's saying is, just because your mom has blue eyes and brown hair does not mean you're going to inherit blue eyes and brown hair. Those two traits are inherited separately. Maybe you'll get them, maybe you won't, because brown hair has no influence on blue eyes and blue eyes has no influence on brown hair. Okay? So, why do we care? Why does that matter? Well, believe it or not, you can actually study the probability of acquiring 2 different traits. So you could study the probability of getting brown hair and blue eyes. But it's kind of crazy. It takes a 16 square Punnett square. Looks something like that. Because you have to come up with all the possible gametes and cross them and then read your Punnett square. Now, that's something we would dig deeper in if we were involved in one of our on-demand videos. But I will give you a hint. For dihybrid crosses, if it's 2 heterozygous individuals, our phenotype will always be a ratio, and they often report these in ratios of 9, 3, 3, 1. Okay? All right, I got a question. Genotype would be the alleles, yes, and phenotype is the characteristics, yes. Great question and great clarification. All right, so Mendel did all of his studies in the 1800s. We've learned a lot about genetics since then. So now we've got to look at some of the other ways that traits are inherited, because most traits actually aren't inherited like Mendel predicted, but many are. And so we were going to analyze what is referred to as non-Mendelian inheritance. Oh, and I've got stuff on here from past. We don't want that. Oops. Let's get those off of there. Oh, I guess they're staying. My bad. Sorry, guys. All right. So of the non-Mendel inheritance, we have 4 that we're going to shine the spotlight on. One is known as incomplete dominance, codominance, multiple allele, X-linked or Y-linked. And they sometimes call this one sex-linked because it's linked to the X chromosome typically. Y is more rare. So let's take a look. Excuse me for a minute. Okay, so incomplete dominance. Incomplete dominance is a type of inheritance where the traits are blended. So, for example, now my illustration's on their work. Snapdragon flowers. If you take a white snapdragon flower and you cross it with a red snapdragon flower, all of the offspring will be pink. So when you think of incomplete dominance, think of the word blend. Like when you were in elementary school, if you mixed white and red paint, you got pink. Incomplete blend. Codominance is both versions of the trait will show up. Neither one of them is recessive in any capacity, and they are both boldly going to show up. This is the beautiful example. It is a checkered chicken. That's what they call it. I love it. I love saying that. I don't know why. The checkered chicken feathers are not gray. They are black and white. Both traits show up. So in this particular species of chicken, if you breed the black version with the white version, all of their offspring will be checkered. Okay. Now, a way to remember this one is codominance. Co-workers both show up to work. So both of these traits are showing up. Okay. All right. So let's take a look at our next one. Multiple allele. This is where more than a single allele pair determines the phenotype. So far we've been talking about like big B, little b, but in this case, it's more than just one pair of alleles. An example of that is blood type. Blood type actually has a gene that they refer to as I that then A or B sit on, and those alleles together determine our blood type. Then finally, the sex-linked version. This is where the trait is carried on a sex chromosome, and it is typically the X chromosome. Rarely is it the Y. And if you think about it, that makes sense, because if the trait was carried on the Y, then only males would get it. It would fall more in line with like diseases, because if it was an important trait, women would probably need that trait too. But this is impactful mainly to men, because remember that men, for a sex chromosome, have an X and a Y. So remember that women's sex chromosome that makes us female is XX. Male's sex chromosome is XY. So they only have one opportunity for this trait. If they get a recessive, then they have the recessive condition. Where women have two opportunities for the trait. So let's say she has the recessive trait here, but if she has the dominant trait right there, it overpowers it and she has the dominant condition. So the woman has twice as many opportunities to get a positive, or not a positive, a dominant trait, where the male only has just that one X and that one opportunity. So what happens here then is conditions that sit on the X chromosome, males have at a greater rate than females have. An example of that is color blindness, red-green color blindness. So if you can see the numbers in there, you're not red-green color blind. But if you're male and you can't see that in there, it's not a surprise because you have less chance or a greater chance to get red-green color blindness. For those of you who are dying to know, it's the number 74 that's in there. Okay. All right, folks, you guys have been amazing. That was our content for today. So I always like to bring you back to our resources so that if you missed the QR codes the first time, you can use them now. If you have an iPhone and you take a screenshot, those QR codes will actually work. If you have an Android, you would need to access those addresses, those email addresses that are on there. But at least you would have this resource at your fingertips. Remember that you can bundle, and bundling will give you everything you need and save you money. So SurePass Pro, the only difference between it and SurePass is that you get this 1 hour of private tutoring. And speaking of 1 hour of private tutoring, I used to be in the classroom and I miss seeing my students. So if you are interested in an hour of tutoring, please reach out because I would love to work with people one-on-one. I get great reward out of that. That is one thing I do miss from the classroom. So if you're thinking you really need that, I am eager to be your tutor. I can do your chemistry, your biology, your anatomy and physiology, and your scientific reasoning tutoring. I got you covered. And of course, you can see Tammy for English and reading and Brandon for math. They would be happy to work with you as well. And there we are again. There are those QR codes so that you can just scan those. Again, take your screenshot and that would work for your iPhone, or just reach out to archerreview.link/TEAS tutors. And then finally, I would so love some feedback. How am I doing? I would love to hear that. So I'm going to leave that up for a little bit, but a friendly reminder, this was content. Tomorrow is our practice test. So come back. Let's see how much you retained. Let's see where your strengths are. And we'll have a great afternoon tomorrow as well. I'm going to leave that up for a little bit, but otherwise, you guys take care, and I will see you tomorrow, same time.